Hello my dear students, welcome to Shohini's Bio book. I have completed the basic things about the chapter that is fundamental unit of life, and that is the cell. Now I will be discussing some important points regarding all the cell organelles that you have studied and some basic questions that can come from this chapter, and something that will enhance your learning and that will more clear your concepts. So now some typical notes on the different organelles. Let's go to the first one, that is a cell membrane. I have noted down the basic characters, the main important characters of the organelles that you have to remember. First is cell membrane, which is also known as plasma membrane. I have taught you all of the points that I have noted here. But still, if you have any confusion, let me know. So the characters that are important for cell membrane are selectively permeable membrane surrounding the cell. Sometimes in some books it is also written as semi-permeable to make it easier to remember, but it is better to write down selectively permeable because that is more typical. Okay. It controls movement of substances in and out of the cell, made of lipid bilayer with embedded protein. This is the composition. Now the cell wall, rigid outer layer found in plant cells, not in animal cells. That is the highlighted point to be remembered. Made of cellulose, provides shape and protection. Now remember one thing, cellulose is the component of cell wall in case of all the higher plants. Now some lower organisms like bacteria, they also possess cell wall, but their composition is different from the plant cell wall. Okay, so in case of plant cell wall, it is made of cellulose. It is fully permeable, it has to be. Cytoplasm, jelly-like substance filling the cell, holds all organelles and helps in their movement, site of many metabolic reactions. Nucleus, control center of the cell containing DNA, also called director or brain of the cell. Surrounded by a nuclear membrane, contains nucleolus and chromatin material, which is that is the DNA and protein. Endoplasmic reticulum, in short ER, this is network of membranes in the cytoplasm. I have told you that these are basically the extensions of nuclear membrane. Rough ER has ribosomes, that is why it is rough, and it helps in protein synthesis. Why does it help in protein synthesis? Because the presence of ribosomes help in the work, and ribosomes function is to synthesize protein. Smooth ER, no ribosomes, helps in lipid synthesis and detoxification. That means the removal of toxic substances from the cell. So they accumulate the toxic substances inside them. Ribosomes, tiny organelles, either floating in cytoplasm or attached to rough ER. Site of protein synthesis, present in both prokaryotes and eukaryotes. I have already told about what prokaryotes and what eukaryotes mean. In case of prokaryotes, ribosome is of 7-ase type. In case of eukaryote, it's of 8-ase type. Golgi apparatus, Golgi body, stack of membrane bound sacs, modifies, sorts and packages proteins and lipids. You already know it. Involved in secretion and transport within the cell, known as dictyosomes in plant cells. Mitochondria, powerhouse of the cell, performs cellular respiration to produce energy, that is ATP. Now what does it mean by cellular respiration? So the breakdown of the food materials that we eat is the process of cellular respiration. So in the process of cellular respiration, that is in the process of breaking down the food materials we digest, the energy is released by the mitochondria. So this is the cellular respiration. It has a double membrane and its own DNA. Lysosomes contain digestive enzymes, break down waste materials and old cell parts called suicidal bags of the cell. I have already told you why is it is called suicidal bags of the cell. I have already told you why it is called suicidal bags of the cell. Plastids, it is already known to you that plastids are present exclusively in case of plant cells. Now they are of three types and three of them have three different functions. Chloroplasts, chromoplasts, leucoplasts. Chloroplasts contain chlorophyll for photosynthesis. Chromoplasts contain colored pigments other than green. They may be carotene, xanthophyll or any other thing. Leucoplasts store starch, oils or proteins, and the leucoplast's differentiation is also known to you. Vacuole storage sacs for water, food, waste, large and central in case of plant cells, small in case of animal cells. Help maintain turgidity in plant cells because they store water and they help to maintain the turgidity in case of plant cells. Centrosome and centrioles, that is only in case of animal cells, help in cell division by forming spindle fibers present near the nucleus in animal cells. Now, some differences and some important questions are listed here. First difference, I have written the differences through points one by one. You can write it by differentiating the two columns. That is, in one column you can write that about prokaryotic cell and in the other column that is about eukaryotic cell. So prokaryotic cell, no nucleus, that means no structured nucleus or no definite nucleus or no developed nucleus. In case of eukaryotic cell, true nucleus is present. So no nucleus means no true nucleus. And in case of prokaryotic cell, there is no membrane bound organelles present like that mitochondria, plastid. And in case of eukaryotic cell, membrane bound organelles are present. Prokaryotic cell unicellular mostly, eukaryotic cells are unicellular or multicellular. That means actually prokaryotic cell is only one cell, but when we talk about prokaryotic organisms, those who possess prokaryotic cell are known as prokaryotes or prokaryotic organisms. So normally prokaryotic organisms have only one cell, so that is their unicellular. But in case of organisms who possess eukaryotic cells, that means the organisms who are known as eukaryotes, they may be unicellular or multicellular. So if the difference is asked between prokaryotes and eukaryotes, you should write this point. If it is about the difference between the cell, you can omit this point. Prokaryotic cell is mostly smaller in size in comparison to eukaryotic ones. Now plant cell versus animal cell, you already know the differences I have taught you. That is the difference of cell wall, difference of having chloroplasts, about the position and nature of vacuole, about the presence of centrosome. Plastid versus centrosome. Plastid is found in plant cells, centrosome is found in animal cells. Their functions are different, their types are different. Okay. Cell wall versus cell membrane. Cell wall present only in plant cells, cell membrane present in all cells. And cell wall is rigid, made of cellulose, it is fully permeable. Cell membrane flexible, made of lipid protein, and it is semi-permeable, or you can say selectively permeable. Now do not make the mistake of writing that cell membrane is present in animals. So, it is present in case of plant cells too. Exosmosis versus endosmosis. You know what exosmosis is? When water moves out of the cell, it is known as exosmosis. So when will it happen? When one cell will be placed in hypertonic solution, then the water will move out of the cell. Then the phenomenon will be called exosmosis, and what it will result in? It will result in the shrinkage of the cell. So these are the all points that's noted here. Go through it. Now some questions can be asked, some tricky questions. What will happen if there is no cell wall in plant cell? So you have to answer accordingly. You have to think about the function and importance of cell wall in case of cell. Then you have to answer what will happen if there is cell wall absent. So without a cell wall, the plant cell would lose its rigid structure and be more vulnerable to osmotic pressure, because the osmotic pressure will be severe and it will help the cell to burst. So it will not help the cell to keep its rigid and definite shape and structure. What will happen in the absence of mitochondria? Now remember, what is the function of mitochondria? Mitochondria's function is to produce energy that will help every organism to do their basic daily works. So cells would not be able to efficiently produce energy, leading to impaired cell functions and survival. Why do muscle cells possess more mitochondria? Muscle cells require more energy for contraction and movement. So muscle cells are most active cells of our body. That's why they have more mitochondria to generate more energy. Who helps in cell division in plant cells? Because plant cells do not have the centrosomes. In plant cells, cell division is managed by microtubule organizing centers, or you can simply write microtubules. Why is mitochondria known as a semi-autonomous organelle? Mitochondria have their own DNA and ribosomes, allowing them to produce some of their own proteins and replicate independently. I have already taught you this. Now give a practical example of exosmosis. Resins, you have all seen the resins. If they are placed in concentrated sugar solution, they will lose water due to exosmosis and will shrink. So this is a practical example that we all can perform. Now, I have told you about the types of ribosomes in case of prokaryotes and eukaryotes. It is of 70s, 80s types. So what does XS mean? Let's go through that. So what does S mean in case of 70S and 80S ribosomes? So S stands for Weidberg unit. The S stands for Weidberg units, after the name of the scientist. Okay, which are a measure of the rate of sedimentation during centrifugation. Basically, centrifuge is a machine that differentiates the cell organelles depending upon their size and composition. So this is a rate of sedimentation. How fast they sediments, how often the sediments, the layer of sedimentation formed during centrifugation. We measure the rate of sedimentation through this unit. So these are the Weidberg units. So in case of 70s ribosomes, the measurement, the rate of sedimentation is 70. Now you do not have to go to deeper concept, just remember the basic things. Okay, so this is related to the size and shape of the ribosome and its subunits. The Weidberg unit reflects how quickly particles sediment in a centrifugal field and is not directly proportional to size. So it doesn't mean that the larger particles will have a greater sedimentation rate. It is not like that, but rather to the overall density and shape of the particle. It depends upon the density, the composition, the materials which are present in that particle, the shape of the particle, and many more. Now in case of 70s ribosome, it is found in prokaryotes like bacteria, and the ribosome has two subunits, we'll know. So the subunits of 70s ribosomes has a sedimentation rate of 50s and 30s. You cannot just plus it and say 50 plus 30 equals to 80. So how is it 70? It's not. When you combine the particles, the sedimentation rate of the total formation might change. And the 80s ribosome found in eukaryotes, it consists of 60s and 40s subunits. So you have just to remember them. So the difference in the Weidberg values reflects the difference in the structure and size of the ribosomal subunits in prokaryotes and eukaryotes. So this is all about some important points and type of questions. And there are also some diagrammatic questions, like you have to practice the diagram of mitochondria and nucleus very well. These are the very important diagrams that are often asked to draw and point. So you have to practice them from your book. You can practice them from my teaching sessions. And if you have any question regarding to any points related to this chapter, you may comment it down. I will help you understand the concept and clear your confusion. So thank you so much for watching. If you like this session, please subscribe, follow and share to all your friends so that they will get the benefited also, and comment down below if you want more such lectures on the chapters left in your book. Thank you.